and welcome to the special seminar series organized by the McGill-Douglas Max Planck Institute of Psychiatry International Collaborative Initiative in Adversity and Mental Health, or AMH for short. My name is Germaine, and I'm the AMH coordinator. As the name suggests, the AMH is an initiative established between McGill and MPI that fosters collaborative research activities with the aim to further our mechanistic understanding of how early life adversity is biologically embedded and how this increases the risk for psychiatric disorders. To learn more about the initiative, please visit our website or send me an email at coordinator@adversitymentalhealth.com. I would also like to take this opportunity to remind you that this session is being recorded. Please send an email to the same address if you would like to access the recording. One last note, as I mentioned, please use the Q&A function to submit your questions to the speaker, and Dr. Gotlib has offered that to interrupt whenever you want, and I will ask your question if you have a pressing one to have answered. So without further ado, I will now pass the mic over to Cecilia Flores, Professor of Psychiatry at McGill University.
Good morning, everyone, and I guess good evening for those people in Europe. So I am truly honored to introduce Dr. Ian Gotlib as the speaker of the Adversity and Mental Health Special Seminar. He's the Marjorie Mhoon Fair Professor in the Department of Psychology at Stanford and the director of the Stanford Neurodevelopment, Affect and Psychopathology, SNAP laboratory. Dr. Gotlib is very active in clinical translational research. His discoveries, including his work in neural, cognitive, social, endocrinological, and genetic processes underlying the link between early life stress and depression across adolescence, have made a major contribution to our understanding and current approaches regarding the psychobiological basis of mood disorders. I am proud to say that Dr. Gotlib received his undergraduate and graduate degrees here in Canada. He completed his bachelor's in psychology at the University of Western Ontario and received his master's and PhD degree in clinical psychology at the University of Waterloo. He began his academic career, I think, right after finishing his graduate school at the University of Western Ontario, serving as professor of psychology and of psychiatry for a number of years. He was recruited at Northwestern University as director of clinical training and worked there for a few years before being recruited at Stanford in 1996 as professor of psychology, and where he also served as a chair of the Department of Psychology for 6 years. I was wondering whether the Canadian cold winters that we have, for example, the past one, play a role in his decision of moving to Stanford, and I'm jealous. Dr. Gotlib has been the recipient of numerous honors and awards. For example, the Joseph Zubin Award for lifetime research contributions to the understanding of psychopathology, the very prestigious American Psychological Association Award for Distinguished Scientific Contribution, the Association for Psychological Science Distinguished Scientist Award, and the very prestigious National Institute of Mental Health Merit Award, which is given to investigators with stellar records. And these are to name a few. Dr. Gotlib has been the editor and author of several books and book chapters. He edited with Constance Hammond, The Handbook of Depression. This book was released in 2002 and was the winner of the British Medical Association Medical Book Competition. And this book is now in its 3rd edition. Dr. Gotlib has over 500 chapters and peer-reviewed publications covering an impressive wide variety of topics related to mood disorders. His pioneer work on mood disorders have advanced significantly the understanding of attentional and memory biases in the processing of emotional information, the neural substrates of emotional dysfunction in depression and anxiety through his work using images, brain imaging, fMRI, and also the cognitive and biological mechanisms that underlie the development and intergenerational transmission of psychopathology in children and adolescents. Dr. Gotlib, we're very thankful for accepting this invitation to speak on Zoom today. And we're also very grateful for your very important contributions to early adversity and mental health. And welcome.
Cecilia, that may be the nicest introduction I've ever had. Thank you. You're just way too kind. I appreciate that. Thank you so much for inviting me to talk with you today. It's just really an honor for me. I followed your work, feels like forever. That's what happens when we get old, right? That it's just really a privilege for me to talk with you. So what I'd like to do today for a little bit is to talk about our work in depression and early life stress, neurodevelopment with a focus on adolescence. I'm also going to talk about what the pandemic did. We all know what it did to everyone, but what it did to our research. And I think to research in general, if I can at the end, talk about some lingering questions about how we go forward with research. So I'm delighted to take questions. I hate waiting till the end of a talk. So if you have questions, please, if you can put them in the Q&A, and Germaine I've asked her to interrupt me. So thank you. Let me start just a little bit about depression itself. So depression as a psychiatric disorder is the single leading cause of disability worldwide. There are over 300 million people in the world diagnosed with major depressive disorder, far more than with coronary heart disease, diabetes, AIDS. The estimated annual cost of depression is about $500 billion. And that includes direct treatment costs, but also includes absenteeism, lost productivity. Depression is also a recurrent disorder. So over 3/4 of people who develop depression experience recurrent episodes and other lifelong difficulties. So one of the things that we've tried to do is to try to understand how we might be able to prevent that first episode, to see if we can prevent this negative cascade over the life course. I should note that depression is also a significant risk factor for suicide. So a diagnosis of depression is associated with almost a threefold likelihood in the increase in the likelihood of suicide, with 2/3 of victims dying in their first attempt if they've had a diagnosis of depression. I want to focus here on adolescence as a period of risk for depression. A significant proportion of depressive episodes emerge during adolescence. And in fact, about 1/4 of all individuals in the US will have experienced major depressive disorder by age 18. It's just an alarming statistic. Early onset or adolescent onset depression is associated with a more severe clinical course, higher risk for relapse and recurrence. You can see Breslau's graph here showing the spike in depression after about age 14. And these statistics have just gotten worse, leading to headlines like, for 80 years, young Americans have been getting more anxious and depressed. More than 1/3 of teenage girls, 33% experience depression. This, by the way, highlighting the sex difference that we'll talk about a little bit later. Leading the American Pediatric Association to say that all teens should be screened for depression. This increase in suicide rates. So for us, the question has been, how do we understand the emergence of depression in adolescence? Certainly, there are risk factors like a family history of depression, and we've done some work in that area. Being female itself is a significant risk factor for depression. Here today, though, I want to focus on the importance of early life stress as a risk factor for depression. So early life stress, or ELS, is a significant risk factor for the development of major depressive disorder in adolescents. And in fact, it's associated with an odds ratio or an increased likelihood about threefold, just under a threefold increase in risk for depression if you've experienced early life stress. It's also associated with a more persistent course of major depressive disorder, and it hinders response to treatment of depression. So greater exposure to early life stress, a poorer response to intervention. Okay, so how are we to define early life stress? I'll talk a little bit more about exactly the kinds of events that we're looking at, but it's essentially childhood exposure to threatening events like parental divorce, mental illness, abuse and neglect, family and community violence. And I want to broaden our perspective to talk about community neighborhood effects in early stress and depression. I'll do that a little bit later in the talk. We've known for a long time about what people have called ACEs, or adverse childhood experiences. And so there is, can you see my cursor moving? Great, okay.
Yes, we can.
Okay, perfect. That means I can use that. So there is this pyramid, and it's been very clear that exposure to early life stress is associated with greater likelihood of disability, social problems, depression, psychopathology is in here. We know far less about the mechanisms by which early stress leads to these difficulties. And so that's in part what I really want to focus on today is how are we to understand this link between early stress and the experience of psychopathology in adolescence and beyond. So some years ago, we initiated a study of early life stress in adolescents. It was a multi-domain study looking at exposure to early life stress and the emergence of various forms of psychopathology, including depression in adolescence, but with a real focus on trying to understand mechanisms that might underlie that association. So we recruited a cohort of 200 healthy boys and girls who were exposed to a range and severity of early life stress. We started that before ABCD. We thought this was a big sample. And then of course, ABCD is in the 10, 12, 14,000. I like to think of ABCD as the Titanic and we're kind of a catamaran. And so we've been actually able to pivot a few times in ways that I'll describe to you, still with a reasonable sample that allows us to assess some of the questions that we were interested in. So it's a longitudinal study. We started conducting comprehensive assessments of these children 2 years apart. So at first ages 9 to 12, 2 years later, ages 11 to 14. Then we had, that was a NIH grant. It was renewed for another 2 assessments. That is another 4 years of assessments. And we had started time 3 when they were ages 13 to 16. When the COVID shutdown here happened in March of 2020, so 2 years ago, we were about 2/3 of the way through T3, and we're just starting the 4th wave of assessments for some of the children, the adolescents. And I'll talk a little bit about more about COVID and what that did and is doing to our assessment. But I want to show you, I want to overlay our assessments on kind of the prevalence of depression over adolescence. And you can see that our first 2 assessments, the time 1 and time 2 assessments, are early on in the emergence of depression. It's really in the time 3 and time 4 assessments that we're trying to really get a sense now of what are the mechanisms underlying this increase in depression and suicidal ideation, in suicide attempts. We are continuing T4 now. So I'll talk about some of those data in a few minutes, and we're developing a T adult, which would be a T5 assessment that we can continue to try to understand some of what's happening here. So what did we do? We interviewed all of the children and their parents about a wide range of stressful experiences. It was probably a 2-hour interview about life stress. We had then a panel of raters code each identified event for objective severity. So we didn't rely on the child's or adolescent's or parents' reports of severity of event. We did something as Brown and Harris did in the Camberwell family interview in England. We then took scores for each type of event. We summed them to create a kind of severity severity score for ELS that represents the complexity, the severity, the timing. We've done more sophisticated analyses of these with different scores for deprivation, for unpredictability, for threat, that I'm happy to talk about a little later as well. To give you a sense of the kinds of events that children, adolescents in our sample experienced, so ranging from witnessing severe illnesses, family members moving in and out, seeing war, terrorism. Again, this was probably 2015 when we started doing this, 2014. So death, family illness, physical assault was relatively rare. Kidnapping, witnessing sexual assault was relatively rare, but we have a good distribution of exposure to early stress in this sample.
Dr. Gotlib, I have to interrupt you. There's a question from Alice. It's what about some studies suggesting that adolescence extends to 25 years old? And what age are you considering to do adults?
So I am fine with whatever you want to call adolescence. We didn't tie our definition to our age range, so I'm just going to go back and say, so we have T1, T2, T3, T4. We've always run into a, I should also say before I answer that we did not match boys and girls on age. We matched boys and girls on pubertal status. So there are age differences at each age, at each pubertal status, right?
Actually, Alice just mentioned, so she's referring to the potential T5 is what.
Oh, yeah, so thank you for that. And if you'd like to join our lab and talk about the T5, we're developing that protocol now. Again, I don't care a lot about labels. So I'm fine with calling them late adolescents. I'm fine with calling them young adults. It's not really going to change what we do as we follow some of these people through. So I appreciate the question that you're asking. I'm just going to call them, let's just call them T5s. And I won't talk about that because we haven't started that yet. They're now starting to be 20. And that's driven by the next renewal being due in November. So we have a little bit of time to get some pilot data for that protocol. So email me. I'm happy to hear thoughts. And thank you for asking. And please, any other questions, I'm happy to to give just as strong an answer to those questions as I did to this. So ELS, let me just show you, we've been replicating kind of what we already knew, that early stress is associated with increases. Here you can see in both males and females, greater severity of ELS associated with greater internalizing problems, greater externalizing problems. You can see here the females having a a stronger association between ELS and internalizing than males, which is what you'd expect. So this is what we found this. These are our data. This is what other people have been finding as well. I want to come back though here to say why. So what is it about adverse experiences in childhood or early life stress that's leading to these difficulties in internalizing or externalizing behaviors? So, excuse me, cough, not COVID. So these are the some of the mechanisms that we've been studying in our sample. And we've been fortunate to be able to have a sample where they've been open to allowing us to go back to, so we have, you know, you don't need to know this, but T-mid, so interim assessments in between these 2 years when we think we should be looking at this. So we've been looking at cognitive functioning, brain structure, function, connectivity, pubertal status. I mentioned that we equated boys and girls for pubertal status at the beginning of the study and looked at sex hormones. We've been looking at biological aging, and I'll talk about that. I think that's been a really interesting finding to come out of what we've been studying. Stress hormones, cortisol, inflammation. I'm also going to talk about neighborhood disadvantage. So I have a postdoc, Jonas Miller, in my lab who got interested in kind of broader SES neighborhood data. And if you have participants' addresses, you can actually go back, and we have data from California Environment on air pollution, water pollution indices, SES, neighborhood level SES. So we've been able to go back, get those data, and integrate them with the data that we've been collecting directly from participants. So I'll talk a little bit about that. EMA from smartwatches and actigraphs and sleep. So I'm just gonna share a little bit of the kinds of data we've been collecting, the kinds of results we've been getting, and how we've been trying to move forward with this. So let me just quickly show you, early in my career, we started studying cognitive biases in depression. We used a dot probe task that many of you may be familiar with. We hadn't done that in the context of ELS before. And with Kate Humphreys, what we found is that early life stress is associated with a strong avoidance of fear faces. The greater the exposure to early life stress, the stronger the avoidance to fear faces. And it looks like that attentional bias is actually mediating the response between ELS and subsequent social problems. So not surprisingly, there's a cognitive bias here that's been associated with both ELS and with social problems. It turns out that when you look at amygdala activation as a function of early stress, you can see this greater exposure to early stress, greater amygdala activation to these subliminal fear faces. And you tie this to the avoidance, and you've got this immediate attention with a subsequent avoidance leading to social problems later on. So just one example how we've been starting to integrate cognitive data, neural data. Kate Humphreys also started looking at hippocampal volume as a function of early life stress. Kate did a really nice job of separating stressors that occurred early in life, that is before age 6, and stressors that occurred later in life between age 6 and age 9 to 12 when they entered the study. And what she found is that it's really the early stress what seems to be this sensitive or critical period of exposure to early stress that's associated with smaller hippocampal volume at the T1 assessment, that is at ages 9 to 12. Okay, so we're getting a stress biology story here, and we pushed the biology story to look at a construct of biological aging. I know some of you are familiar with this. It's this interesting concept. Natalie Kolich, who's now working with Kate McLaughlin at Harvard. Natalie was a graduate student in my lab, studied telomeres in the context of risk for depression, wrote this really nice review of biological aging following threat and deprivation, which are the kind of types of ELS that Kate McLaughlin looks at. And Natalie found that early life stress is associated with accelerated aging. And so what does that mean? Well, it means that at the same chronological age, individuals can differ in their metrics of biological aging. And I'll show you exactly what that means in our sample. So we have pubertal stage data in our study. And what you can find, what we found here, what you can see is that for females, the greater exposure they had to ELS, the greater their pubertal stage was at the same age. So girls at the same age who were exposed to early life stress were in more advanced pubertal Tanner stage, okay? We also find more mature neurophenotypes, that is a lower amygdala ventral medial prefrontal cortex connectivity, as a function of greater exposure to ELS. So faster neural development. And what you can see here is also earlier age of menarche, right? So with greater cumulative stress, earlier age of menarche for girls. What does that mean? Why is that important? Well, it turns out that that earlier age of menarche, that is earlier puberty, predicts greater internalizing problems 2 years later. So we've got what seems to be faster biological aging in individuals who were exposed to early life stress. I'm just going to
another interruption, Dr. Gotlib.
Yeah, please.
Sorry, another question from Alice. Have you seen associations with SES or ELS with global volumes such as gray matter or TBV?
We haven't seen. I'm going to talk about the environment, and so we haven't seen global volumes, but we've absolutely seen connectivity differences, activation differences, and structural volume difference, but not whole brain, which is an interesting, we haven't focused on that. We should. Jonas with Emily Dennis has been looking at tensor-based morphometry, that is expansion and contraction of different brain regions as a function of early stress, and they found that, but not global gray matter. So interesting, and I'm happy to talk with you about that. We have the data. If you're interested, send me an email and we can think about looking at that.
Now, another question, have you seen associations with cortical thickness?
We have seen associations with cortical thickness with risk for depression, but not as a function of early life stress. So young girls, and I think it's sex specific, young girls who are at familial risk for depression, that is daughters of depressed mothers, without ever having been depressed themselves, have thinner cortices, that is lower cortical thickness, than do girls without a family history of depression. We haven't seen that in our early life stress data, but I want to say we haven't focused on that yet. So it's a great question, and I will make a note of that. I'm not going to complain about too much data in too little time, but I'm going to complain about too much data in too little time. So email me if you're interested in playing with us. Thank you for the questions, and I'm happy to continue. We're in good time. It's only 8:30 and we're in good time. I mean, sorry, my time. So we have lots of time. I'm happy to take more questions. I want to broaden this a little bit. I mentioned that Jonas Miller, and it's wonderful to have smart postdocs and grad students in your lab. I mean, I just kind of let them do what they do, and they come up with interests and ideas. And Jonas got interested in neighborhood and the environment. And we were able to, with the addresses that we have of our 200 participants, compute indices of neighborhood disadvantage. And you can see here in our sample that neighborhood disadvantage, the greater exposure to early life stress, the higher the neighborhood disadvantage. And it's really a cumulative summative metric of education, income, housing. And in general, let me just say that it's not just SES, that it's, for our data, it seems to be much more nuanced than that, reflecting social inequality in the way that neighborhoods are formed and analyzed. And what we're finding is that adolescents from disadvantaged neighborhoods, this biological aging that I've been showing you is associated with ELS, we're also getting indices of biological aging moving with neighborhood disadvantage. And I'm going to show you some data for telomere length and neurobiological data. So kids who are in neighborhoods characterized by greater disadvantage, that is 1 standard deviation above the mean, at each age have shorter telomere length. So telomere length, we've, with Natalie Kolich, I mentioned earlier, found that daughters of depressed moms the daughters are 10 to 14 years old, already have shorter telomeres, it's a measure of biological aging, than their peers, non-depressed daughters of non-depressed mothers. Telomere length, Liz Blackburn at UCSF won the Nobel Prize for her studies of telomere length, predicts coronary heart disease, predicts premature death. So again, these are young kids, but already with increasing neighborhood disadvantage, they're showing shorter telomere length. They're also showing that same stronger amygdala VMPFC functional connectivity, that brain connectivity metric of advanced biological aging, more mature neurophenotype here. So what we're finding is that neighborhood disadvantage is seeming to have this strong effect on accelerating biological aging, even in children and adolescents. You shouldn't know, I don't want you to memorize this table, but I do want you to see just the different ways that you can start to integrate some of these data. So Jonas Miller was interested in particulate matter, 2.5. These are very small particulates. It's a measure of air pollution. And what he's finding is that early life stress matters in the effects of particulate matter on brain development in adolescents. And it looks like high and low ELS makes a difference for whether particulate matter, air pollution, has an adverse effect on brain development, right? And so we need to do a lot more work here. These are not findings to look at for you other than to know this is significant, right? And to the effect it locks into climate change here in California, it locks into wildfires and smoke, and which is where a lot of the particulate matter data are coming from California. So these are real findings, and it looks like an experience of early stress is making things worse, is setting kids up for a more negative cascade of effects of air quality. We've also found water quality on brain development. So, Germaine, questions? You're muted.
Oh, I thought I pushed the unmute. We have a question. Yes, the question is, the neighborhood disadvantage is interesting. I wonder if there might be contributions, correlations with factors such as nutrition, genetics, epigenetics?
Yeah, without a doubt. And thank you for that. So we're meeting, I'm in Vancouver next week for a meeting with Mike Kobor at UBC. We've been doing some mitochondrial DNA, some methylation work with Mike on this sample. And it's absolutely the case that these are going to tie together. We haven't tied that to the neighborhood data yet, but we have tied it to early life stress. And early stress is, it's a whole other talk, but it's part of the metric of biological aging. So I think you're absolutely right. Genetics, we just don't have the sample size to start to look at, way back in a former life when we were looking at the serotonin transporter gene, when that was okay to do, when it was okay to look at single nucleotides, candidate genes. We found some interesting data with depression and 5-HT transporter gene, serotonin transporter gene. I think we don't have the data. They do probably in ABCD to look at genetics. I think epigenetics with neighborhood data is absolutely going to yield findings. I don't know that that we have the power to look at GWAS, larger sample are going to be needed for that. I think it's a great question. Thanks. So I don't know how you all are with COVID-19. Cases here are moving back up. We had been shut down. I'll talk a little bit about that. I teach here at Stanford. I've been teaching in a mask to students in masks. After teaching remotely, the teacher doesn't have to wear a mask. Students still need to wear masks. We're going to be back into mask mandates. That's my prediction, given what's happening to cases. COVID-19 and mental health has just been horrible for anxiety, depression. You see these kinds of, particularly with kids, teens, you see headlines like this all the time now, to say nothing of scientific papers. So I want to talk a little bit about what COVID-19 at a very kind of personalized way did to our research. So as I mentioned, we had this early life stress study, longitudinal data have been going on, T1, T2, T3. We were forced to shut down at Stanford all in-person research in about March, April of 2020. We were shut down here, and that meant we had to stop our data collection. And our data collection was scanning and cortisol and saliva and inflammation and blood. And so we developed an online protocol survey to give to our participants, including a coronavirus impact survey and some other self-report measures that we gave to our kids. And I'll come back to this, but understand, so we had done in-person pre-pandemic, time 1, time 2, about 2/3 of time 3, and a couple of kids in time 4. So in the survey, what do we find? So here are the data from our kids internalizing data, severity of internalizing problems, pre-COVID, post-COVID. I mean, just an enormous increase. This is kind of past 2 weeks, but this is maybe 2 to 3 months into the shutdown. And we're seeing this increase in internalizing symptoms. We also, I want to show you this. It's related to their baseline severity of early life stress. So those kids who had experienced greater early life stress pre-COVID had higher symptom levels of depression, higher depressive symptoms during the shutdown, and that seemed to be mediated by how much stress they were experiencing during the shutdown. So we had been trying to analyze some of our data in the Life Gives You Lemons, Try to Make Lemonade perspective. I had postdocs and graduate students also shut down, also working remotely. So we tried to use as much of our time 1, time 2 data, for which we had complete data on these kids, to predict who was doing better, who was doing worse during the shutdown, okay? So I'm just gonna present just a couple of findings to give you a sense of what's happening. So Jonas Miller was able to use, we had heart rate variability data from a Trier that we had given these kids at time 1 and time 2. And what he's shown, ELS was related to heart rate variability, greater ELS, greater heart rate variability. I just want to show you that heart rate variability is actually moderating the relation between COVID and emotional problems. So that COVID stress seems to be really important. How much stress these kids are experiencing during COVID is really predicting greater emotional problems during COVID, but even more so moderated by pre-existing, what we would call, if we were an insurance company, a pre-existing condition of higher resting heart rate variability. I want to focus just for a moment on this concept of resilience, because I don't want this to get lost. I think this is really important. So some kids did worse. Of course, all these headlines, lots of kids did very badly during the pandemic. But there's a subset of kids who actually were doing fine, who seemed to be doing okay, who seemed to be what we would call resilient. That is not just the flip side of psychopathology, but more specifically kind of who had experienced early stress, but who weren't showing the emotional difficulties, particularly during COVID, that other kids were. And so we wanted to see if we could identify factors pre-COVID that would predict who did better during the pandemic, who was resilient. And so Raj Chahal, who was a postdoc in my lab, really focused on this executive control network connectivity. So we had resting state data from all of our participants, and she was interested in executive functioning, those kids who were cognitively, whoops, that wasn't me, those kids who were cognitively better and able to navigate challenges. And what she was thinking is during periods of stress like COVID, then higher executive functioning, reflected in higher executive control network connectivity. And that's really kind of these frontal parietal regions with some posterior parietal cortices. And what she found is that higher... Oh, yes.
Sorry, I don't know if this was actually just related right now, but
okay.
Someone asked, have you looked at the developmental impact of mindfulness-based interventions?
No, and I'm happy to talk more about that in the discussion. We have not done any interventions with these kids. It's always a tension because this is a naturalistic study of the impact of stress, and as naturalistic as the COVID pandemic can make this be, but we have not done an intervention at all with any of these kids. So I have no idea what mindfulness would be doing. I have thoughts, but my thoughts are no more valuable than your thoughts, right? They're only speculation. So I'm happy to come back to what we might want to do here. But we have not done any interventions with these kids. So I'm hoping that's helpful. If not for the kids, for the answer. Okay. So what Raj Chahal found is that those kids who at time one had higher coherence in that network. So we have this executive control network, and it's essentially higher correlations among these structures within the executive control network, tighter coherence. Those kids were buffered, right? That's this line here. They were buffered against the effects of COVID on internalizing symptoms that kids with lower coherence experienced, okay? So I understand it's a more complicated story, but it does look like higher coherence in the executive control network is protective, and it's protecting those kids from experiencing the normative increases in internalizing problems that kids with lower coherence are experiencing as they go through puberty. Okay. We're at 8:50. I don't have much more, but I have something that's really been bothering me. And so this is the first time I've shared it with anyone outside the lab, and I want to share it with you. So we're back. We're scanning kids. We started scanning kids in December of 2020. We were one of the first labs to resume scanning because we had a longitudinal study and they knew that we were losing data. Our kids refused to stop aging while we weren't scanning them, but we were able to start bringing them back. We've been scanning for just over a year now. So that's fine. So we had a period of 9 months, 10 months where we were shut down. And so we have this interval between time 3 and time 4, time 2 and time 3 for some of our kids. That's a longer interval than pre-pandemic when we were going time 2 to time 3, 2 years, okay? And of course, you can just use you can just use interval between assessments as a covariate, and maybe you're fine. But it just bothered me that kids that we scanned after the pandemic may not be the same as kids that we scanned before the pandemic. And it took me 3 lab meetings with my group to figure out how to express this. And I'm probably not going to express it well here, but I want to try. So this is not a question of what has the pandemic done within person. That is, how do you assess, as I did, I'm going to just go back here. Whoops. How do you assess this, right? I'm not talking about this change from pre-pandemic to post-pandemic. what I'm trying to get at is, our 15-year-olds that we're now scanning in T3 or T4 or 16-year-olds, are they the same as the 16-year-olds that we scanned at T3 before the pandemic? Okay, they're different kids. So it's not really in-person change, and it's not just using the interval between assessments and continuing our longitudinal questions as they were. It's, what if they're different? What if 16-year-olds are just different now after this pandemic? Well, after. Optimistically, I'm calling it post-pandemic, but let's call it peri-pandemic. So 2 years in, are 16-year-olds the same as the 16-year-olds that we scanned or assessed during the pre-pandemic period? I'm rambling. I'm trying to tell you what I struggled with. So here's what we did, and maybe this will help. We took—
Interrupt you. There was one question.
Please.
Because I don't know what the timing, but I think the question is broadly, I was wondering what your thoughts may be on if these early predictors may be transdiagnostic. Are there underlying mechanisms that you think, ELS, neighborhood risk, et cetera, may be impacting that leads to depression, anxiety, or other forms of psychopathology?
Thank you. I think all of these things are transdiagnostic. So this is a whole other discussion. I think depression as a diagnosis is overrated. I think that it's completely non-specific. I think that everything I presented to you in the context of internalizing, externalizing, which makes to me more sense with the kids. I think the heterogeneity of depression is a real problem, but that's a whole other— so take everything I'm saying as these are problems transdiagnostically for poor mental health going forward. And for some kids, it'll look more like depression than anxiety. For other kids, maybe more like anxiety, but that's all within the margins of unreliability of diagnoses. That may not be satisfying, but that's how I feel. So I want to leave you with this. Maybe it's only important to me, but I just can't help, but I just want to tell you what we did. So we took pre-pandemic and post-pandemic assessments. Remember, it's longitudinal, so these are the same kids, but we were able with 200 kids to get a subset of kids that we assessed pre-pandemic and an age-matched subset of different kids in the sample, but at the same age. So 15-year-old kids that we assessed at 15 pre-pandemic, and different kids that we brought in to assess at 15 years old after the pandemic. Does that make sense? Yeah. So we started to look at, do they look the same? If they look the same, then we're great. Then we can just use age interval as a covariate and go ahead. And so I'm just going to show you some of the self-report data. We're still scanning, still looking at the scan data. I know it's a lot of data. Let me just highlight. So here's the youth self-report. So the pre-pandemic sample and the peri or post-pandemic sample. Social problems, look at this, we're fine, right? These two groups of age-matched kids look the same, roughly. But not here, not in depression, and not in the manifest anxiety scale, right? Where 15, 16-year-olds post-pandemic are more symptomatic than 15, 16-year-olds were pre-pandemic. That changes how we do our longitudinal research. We're not the only ones with longitudinal study that got interrupted, right? I think this is an important question going forward. And it's kind of has the pandemic made a permanent change in kids that we can no longer just go on with our longitudinal research using interval. Yeah, it's a longer interval between assessments while we were shut down, but there's more going on here. And I left this in just because I like it. We had, on another study, interviewed a large sample of like 1,000 women who were pregnant during the pandemic. And we also had our babies study of pregnant women pre-pandemic. And I just want to show you their depression scores, right? And it's clear that women during the pandemic who are pregnant are more depressed for lots of reasons than women who were pregnant pre-pandemic. Maybe this helps because these are definitely different women, right? But this is the same issue. And it's what does it mean, right? So I know we're coming to the end. I'm just going to leave this up and just say, look, we're trying to understand mechanisms. I think we've identified some of the large samples with multi-domain data. I think it's just so important for understanding going forward. Cognitive biases, brain function, these don't happen in isolation. They don't happen independent of neighborhood effects. Joan Luby and Deanna Bartsch just published a really nice neighborhood crime effects on neonatal, during pregnancy, of neonatal functional connectivity. So I think there are real opportunities for broadening our assessment of mechanisms to understand how early adversity leads to problems later on. So thank you for bearing with me. It's a weird format, but you all get to just go to bed after this. So thank you very much. And I really appreciate your questions, and I'm happy to stay.
And thank you very much, Dr. Gotlib. Actually, I'm going to allow the participants to ask their questions because I've noted them, and I'm going to open the mic for.
Should I can stop sharing. Does that help?
Yes, that would be.
Great.
I think Julia Feitz has a question. If we can I'll just open it up for her. Okay, go ahead, Julia. Can you ask your question?
Can you hear me?
I can.
Okay, great. So my question is related to the association between the hippocampal volume and early life stress you showed in the first half of your talk. And we are running very similar analyses, or like analysis in a similar direction. And at the moment, we're evaluating which covariate to add to the models. And I was wondering whether in your plots you showed, which had also an interesting sex effect, whether you used intracranial volume as a covariate. And
yeah, always. And
always.
If I may refer you, so Kate Humphreys is the first author. It's a 2018 or 19 developmental science paper. So you can see all of the covariates analyses.
Oh,
but
perfect.
Yes, always use intracranial volume as a covariate.
Okay, great. Thank you. So yeah, I wrote down this paper as well. So I will definitely have a look into it.
Good. Glad you're looking at that. Daughters of depressed mothers, by the way, who before ever experiencing depression themselves also show smaller hippocampal volume. So a lot of these effects are just so non-specific. I think it's a stress, emotional difficulties story at the end of the day.
Okay, thank you very much.
Sure.
Thank you. Next question comes from Dominique Walker. I will open up for Dominique.
Yes, thank you very much, Dr. Gottlieb. This was a fantastic talk. I really enjoyed it a lot. I wanted to ask you a question. I had a question in mid-talk, and then you discussed that you're not doing intervention. But I wanted to just have a sense of how much those markers that you highlighted, both of heart rate variability and the higher executive control network, could, in your view, be a good basis for setting up some meaningful intervention for, especially maybe for kids who have been exposed to ELS maybe later on in their childhood, as you were mentioning, that the effect might be, we might have some more lability in the effect of ELS in terms of intervention there. So I wanted to have your idea about that.
So it's, Dominique, it's a very important question. And I didn't at all mean to say I don't favor intervention. With this sample, we're not intervening because I want to understand the trajectories of the impact of early stress. But the whole point here is exactly what you said, to identify targets for a possible intervention. And so even things like telomere, faster biological aging, there are lots of interventions now that people are using with adults to slow the shortening of telomeres. A lot of it's stress reduction, exercise. So these kind of meditation, stress reduction, coping, I think these are all viable and reasonable candidates for interventions for these kids. And that's the purpose of identifying the mechanisms. Just knowing that early stress leads to depression doesn't give you any sense of what to do about that, right? The stress has happened, they're gonna be depressed, but if we can understand the mechanisms, you can work, and if I think it's a stress story, then to me, that's the target. So certainly mindfulness, meditation, whatever kinds of interventions we have that target these mechanisms, I think are absolutely worth exploring. That's the whole point of what I think we're doing as a field. So
thank you very much. And if I may just have a quick other question, because we've been also seeing the effect of early life stress in the functional connectivity between the amygdala and prefrontal cortex. And I wanted to know if you have looked at the relationship between this, the emotional connectivity and the executive control network.
Yeah, so it's a very good question. So Raj Chahal has, I'm happy to send you some papers, has really tried to understand the effects of different types of diversity on different networks, on the coherence in different networks. It's not a easy or clear story because of course the brain is, we call them networks, but everything's connected, right? So some a little more coherence than others and a little stronger correlations, but everything is connected. I think it's reasonable to start to look at that. One of the explanations for adolescent depression, of course, and risk-taking is faster maturation of limbic than cortical. So not as much a network, but it's the slower maturation of the executive control network. So there should be some distinction, some separation between the executive control network and other, say, the reward network, particularly in adolescence. So I think that looking at the strength of network correlations is going to be important going forward.
Thank you very much.
No, thank you. They're great questions.
Now there's a question from Xinyuan. Can you go ahead?
Sure. Thank you, Dr. Gotlib. Thank you for your so wonderful talk. You talked about early life stress predicted lower functional connectivity between amygdala and VM prefrontal cortex. I was wondering if this pattern or alteration varies with age. And did you examine the structural connectivity between amygdala and prefrontal cortex?
No. It's a very good question. And I want to say we co-varied for age rather than using age as a factor in that model. And I can send you the paper if you send me an email, but I want to say that it's across age ELS is associated, higher ELS is associated with stronger connectivity. So it's not so much an age effect as an ELS effect across age.
Okay, yeah, thank you. Yeah, another quick question. As far as I know, amygdala and the prefrontal cortex, both of them involved in early life stress, and also they are involved in depression. So I was wondering if you ever examine the mediation or connectivity between amygdala and the prefrontal cortex, its mediation or association between early life stress and depression in adolescence.
Yes, it's a very good question. Jonas Miller looked at amygdala connectivity as a mediator from parenting quality to depression in this sample and found a mediation. I don't know that we've looked at the early life stress as the independent variable, but certainly it's a question worth pursuing.
Cool, very cool. Thank you very much.
Thank you.
Yeah, our next question is from Dr. Suzanne King.
Suzanne, how are you?
Hi. Hi, Ian. I hope to see you at SRP this fall.
Yeah, travel is just so much fun, isn't it?
Oh, yeah. So really fantastic talk, and just the body of work, not just this longitudinal study, but everything is so impressive. So thank you for presenting that.
Thank you for saying that.
So I have a bit of a, I don't know if it's anyway, so related to genetics. So I know that you said that, of course, your sample size is maybe too small to look at genetics, but to what extent do you think that the ELS score that you have is a proxy for parental psychopathology? So as I understand it, you may have done, was it Connie Hammond's UCLA stress interview? Is that what you did?
Yes, but about stress for the children. That said, though, we do have measures, self-report measures of psychopathology in the parents. And it's a 0.2-ish correlation. So I don't think it's a proxy for parentals. It's related, right? But it's not. Right.
So for you mentioned George Brown's life events and difficulty schedule.
You're the only other person on the talk who knows that.
who looked at the objective severity of the hardship associated with an event. But George Brown also rated the independence of the event from the person's psychopathology. Right. So, for example, on your list of exposures, you have things like divorce, family financial problems, family legal problems, and so on. So is it possible that children of parents who had those kinds of problems themselves had maybe a lot of these same mechanisms, like functional connectivity, like the telomere length, so to what extent is it possible that the ELS score is a proxy for a family environment that could have heritable aspects, which are also heritable for...
Yeah, I think it's completely, thanks, Suzanne. I think it's completely possible. What we tried to separate was the objective severity of the stressor from the subjective response of the participant to the stress, right? Mm-hmm. So we did that, but it doesn't at all take away the possibility that that kind of transmission of stress, we just took away the subject's response, right, as being part of that stress score. But absolutely, you could be right.
So what if I have two ideas? One is to kind of go through that list of events and see which ones are potentially independent of the parental psychopathology and which are not. But then also, if you were to assess the parents with the same mechanism measures that you're testing the kids, right? So maybe parents with stronger functional connectivity between amygdala and PFC, just genetically, just through heritability, have kids with the same.
Yeah, perfectly appropriate. And if we could scan 200 parents, 400 parents, we'd be right there with you.
Okay. Thanks so much.
Thank you, Suzanne. I'm going to have to drive in a few minutes, but I'm happy to take another question if there is one.
How about we just finish off with Cecilia? I can send out the other questions because there's a few other questions on the chat, but yeah, I can always send them to you, and we can, then you can address them. And
yeah, if okay. And I'm sorry.
Yeah.
I have to.
How about Cecilia? You can have your question, and then we'll.
Yes, thank you. It would be very quick. So
no,
it's really sad to see all the changes that the pandemic has done to adolescents. And just very briefly, if you could comment on what would you suggest would be the balance to deal with the pandemic and at the same time deal with this huge problem?
So I think it's a great question, recognizing the problem for sure. I mean, we're so divided in this country that it had no effect. It's not even a pandemic, it's not a virus, it's like the flu, right? But I think the social isolation during the lockdowns have just been, particularly for adolescents, so critical. And I just think recognizing and offering resources to kids, healthcare here has just been so overwhelmed with the physical, right, aspects of the pandemic that we've neglected all of suicide rates have shot up here for adolescents in addition to depression, anxiety. So it sounds trite, but I think just making sure that kids have access, particularly in schools now that they're back, to resources. But that's easier said than done, right? In a time when we're cutting back on social costs, social funding, right? It's just very, very difficult. And I don't see it getting better before it gets worse.
Yeah, thank you.
No, thank you.
Thank you very much, Cecilia, and thank you very much for this wonderful talk, Dr. Gotlib. And
thank you. And again, I apologize for having to.
No, you stay 15 minutes more with us. That's a lot of time. Thank you so much.
Thank you so much. It's really been a pleasure.
And thank you everyone for attending, and have a great afternoon, morning, or wherever you are.